So we're delighted to have you. Perhaps we'll go ahead and get started. We have a lot of people already on the line. I think others likely to join, but have a lot of ground to cover. So thought maybe we'll just jump right in, given that it's just past the top of the hour. Again, good evening, good afternoon, good morning. It's Mitchell Warren from AVAC. I'm actually here with Stacy Hanna, who leads research engagement at AVAC, and our Coalition to Accelerate and Support Prevention Research Project, CASPR. And I know a lot of our CASPR partners are on. We're delighted on behalf of both AVAC and I'll turn to my dear friend and colleague Ntando Yola in just a minute to welcome you on behalf of APHA, who are co-hosting this webinar. Unique in a number of ways, not least of which in that we don't be surprised if you're on screen, you won't see much because we are not using slides. We really want to make this as much a conversation as possible, although we do have a number of input that we want to provide in the first part of the call and then open it up for discussion and questions. Maybe I'll just do some quick logistics and then I'll turn it over to Ntando also to welcome people. But we'll take a couple of inputs from Linda Gale-Becker, the co-chair of the HVTN 702 trial, from Mary Maravich from the Division of AIDS that sponsored this trial, and she leads HIV vaccine activities there at the Division of AIDS at the NIH. We then have Ntando and other members of both the Vaccine Advocacy Resource Group, the VARG, as well as other community voices in South Africa to give some reflections, and then we'll open it up for questions and conversations. Throughout all of this, if you are on the web feature, you can type in the Q&A at the kind of bottom of the screen. You can type in questions and we'll take them as they come and try to provide a little order to it. You can also email avac@avac.org at any point. We'll put them in the queue, and or you can also use the chat feature. So a number of ways that we really want to hear from everyone. And that's kind of how we'll manage the call. Obviously, this is not the first conversation in the last couple of weeks about this really important trial, nor will it be the last conversation. But we hope it does provide some opportunities for all perspectives to kind of come out on the proverbial table for all of us to grapple with. And maybe before I invite Linda Gale, maybe, Ntando, do you want to say anything on behalf of APHA?
No, thank you very much, Mitchell. And really, again, we would like to welcome everybody. I think as Mitchell has mentioned, and this is really one of the conversations that have taken place, I mean, that are adding to the conversations that are taking place around this important outcome, which really helps us to reflect and think on really how we should be going forward, having received all of this news. And this webinar is also providing, in addition to people who are really coming from leadership of the study, but also so we have a range of community members who really work with communities who have been part of the study, and we'll be introducing them as well after we've heard from the initial speakers. And otherwise, it's really great to have this conversation, and we are really hopeful that it is going to provide us a lot of insights as we go forward in dealing with this outcome. Thank you, Mitchell.
Great, thanks, Ntando. So we'll start, and I think to all of us working in HIV vaccines and HIV generally, our first kind of presenter needs no introduction. Linda Gale-Becker leads the Desmond Tutu HIV Foundation vaccine research activities. She's been involved in pretty much every prevention trial that any of us can think of in the last decade, and of course is also past president of the International AIDS Society. For purposes of today, of her many hats, she was the co-chair of the HVTN 702 protocol, along with Glenda Gray, who is not able to join the call today, but really wanted to first and foremost welcome you, Linda Gale, and also not just you and Glenda, but also Muku and Fatima as your as additional co-chairs of this trial. It takes a village, and it's great that this village was led by 4 remarkable women scientists, and just want to thank you for your leadership and turn it over to you to provide kind of the background and perspective over the last couple of weeks.
Hi, Mitchell and Nintendo. Thank you very much indeed for hosting this, and a great pleasure to share this platform with, of course, Mary and all the many other experts that Nintendo has mentioned, and certainly do want to leave time for numerous voices as we make sense of a time, I guess, perhaps just to acknowledge the sense of disappointment and and a time of reflection, I guess, as we go through that, and then come out the other side ready to take on HIV again and make sure that we do find solutions for South Africa. So I'm going to, on behalf of the three other co-chairs and the protocol team, make some assumptions that people have the basics of the HVTN 702. Obviously, those can be shared, but a number of press statements and other documents have been circulating around the world, so I'm sure that is known. I'm rather going to spend the time just making sure everybody is up to speed with the events that have transpired since the 23rd of January, and then fill in any gaps that people may have in terms of where we are in the process and what the immediate next steps are. So just to step back momentarily to remind everyone that HVTN 702 involves 2 vaccine products, experimental, based on the Thai RV144 trial and conducted in 14 South Africa only sites in a typical one-to-one randomization of active arm to placebo arm. There were 2,700 participants in each arm. And at the time that on the 23rd of January, our DSMB met to reflect on their interim analysis. Now, I just want to make it clear, because I've been asked this a number of times by people, why did the DSMB meet? This was written into the protocol a priori, so in other words, at the time that the protocol was written, that after the 59th infection, this was an infection-driven study, after the 59th HIV infection, every 50 infections thereafter, the DSMB would meet to think about 3 possible options, and many others, but 3 main ones. One being that there was good efficacy, and so you'd want to stop the study because you'd want to roll it out. Or secondly, there was harm and you'd want to stop the study under those circumstances. Or thirdly, if indeed we were not likely to see any efficacy, and therefore you would want to stop the study so as not to waste time and resources and participants' efforts, etc. So this was carefully thought through, that it would happen at a time when there were sufficient infections and sufficient time had passed to be sure that we weren't stopping the study too soon or too late, in fact, finding that sweet spot. So that is an important concept that I just wanted to make sure people understood. And then to say that the 23rd of January, when the DSMB met, they looked at both the blinded and unblinded data and came to the conclusion that sufficient time had passed for them to pronounce that these vaccine products were unefficacious. In other words, they did not prevent HIV, and there was no evidence that this would happen in the future. So again, recall, this is it's a prime boost regime, and there were 6 vaccination opportunities over an 18-month period. So the DSMB met to say that the results of the non-efficacy analysis is that the data has crossed pre-specified boundaries and meets the criteria for non-efficacy. There was no evidence of either increased or decreased infection rates with vaccination. In other words, the vaccine, although not working, is safe and not harmful. And this was also reflected in the fact that the DSMB had met a number of times previously, and we had not seen any safety concerns. Based on this, the DSMB called the Oversight Committee together and made one recommendation and one suggestion. The recommendation was that the study in its active form be stopped for non-efficacy reasons, and that means that vaccinations needed to be halted. But they then went on to suggest that the team should continue follow-up of participants for a period of time that the protocol team would decide on together. Reiterate again, basically a vaccine regime that was safe, not harmful, but also non-efficacious. We obviously galvanized into action on that Thursday of the 23rd. We met numerous times telephonically and in other ways, and we had a number of steps that had to be taken. Obviously, the regulatory body, SAPRA, in the country had to be notified and was done so during that following week. We also started to work on the notification to the participants. First and foremost always is to let participants know the most recent information. And in order to do that, we need to get ethics approval. So a Dear Participant letter was fashioned together with a cell phone communication, an SMS message, which needed to get approval from the various ethics committees that are served by the 14 sites. That meant we needed to let a tight group of site staff know about this, which was done under confidentiality. Wonderful ethics committees approved this in record time. And by Monday morning, very early, Monday the 3rd of February, we started to let participants know via SMS messages and telephone conversations and one-to-one, as well as group meetings at site. Participants were told those two things. One, the vaccine is safe, but non-efficacious, number one, and number two, therefore vaccinations would be stopping, and that we needed people to come to the site for further information. The sites have continued to meet with community advisory boards. That happened at the same time that participants were being advised. And obviously letting 5,400 people know is a huge task. And so many sites had more than 500, 600 people to work through. And so much of that week was taken up just letting our participants know. We also had a series of webinars to let key other stakeholders know. And at 3 o'clock South African time on Monday the 3rd, the announcement went public that the HVTN 702 trial would be terminated early. So that does not mean follow-up will not be conducted. So the study will continue, but without active vaccination. We were able to tell participants that, and as we speak, the protocol is in a very speedy fashion being rejigged and rewritten to include this extra, at this time, 12 months of follow-up for participants who will be invited to reconsent in this new form, and they will be followed up into the future without further vaccination, as mentioned. The reason we're doing this is that there obviously is still much to be learned about this vaccine, a set of vaccines, and it's important that we also continue to follow individuals, because we will continue to keep an eye on safety over a period of time. We are moving as quickly as we can to unblinding, and in fact that started this week. Sites have had lists of their participant assignments sent to them. And this is a process that we do obviously one-to-one. So site visits are scheduled. Individuals are invited back to the site to have a conversation about which arm they were assigned to, and then also a further information given to them about what will be happening with the clinical trial moving forward. So that essentially is where we are at the moment. We have recognized that our communities are very much partners in this. We've had a series of webinars to try to spread the word. There has been a number of announcements over media, radio programs, etc. But clearly we continue to invite the communities most closely associated with the research sites to seek any clarification on what they understand, and also the site staff are working very hard to make sure that the right information is getting to individuals. I'm sure people on the webinar may want to know what the sort of feedback has been like. Again, incredibly humbling that our participants who participated actively in this trial were part of the informed consent process, have by and large fed back that whilst they share our disappointment, they recognize this was experimental. They recognize that this may well have been an outcome. This was always a potential outcome. And so, you know, we continue to be, as I say, humbled by the level of enthusiastic participation in the research endeavor and in this quest to find a vaccine. So we are continually open to hearing any other concerns, but by and large, the feedback has been one of support. And a great deal of enthusiasm to stay engaged in this process. So I'll perhaps stop there and hand back over to you, Mitchell and Ntando.
Linda Gale, thank you so much. That was a remarkably clear description of what I know has been both a huge amount of work and a huge challenge for all of you. So thank you so much. A lot of questions I know coming out, and we'll get to those as quickly as we can. But first, we wanted, in the midst of all of this, to have the slightly larger context as well from the HIV vaccine field writ large. And Mary Maravich, who leads the HIV vaccine program at the Division of AIDS in the National Institute of Allergy and Infectious Diseases of the National Institutes of Health in the United States, that funds the lion's share of biomedical research. And delighted to have you here, Mary, to provide a bit of context in all of this and some reflections as well from someone who's worked across so many aspects of this field and is involved in so many of these areas.
Thank you, Mitchell. I appreciate being invited to participate in this webinar, and I send greetings to all who've dialed in. I did want to echo the points that Linda Gale made, that we are disappointed about the results of this study, and we are very proud of how the study was conducted, and very grateful to all the participants and the staff who conducted the study. I will mention that the DSMB, the Safety Monitoring Board that Linda Gale mentioned, did commend the study staff and the participants for how the study was conducted. It was done flawlessly, and it wasn't the study itself that failed, it was the vaccine. So I think that's kind of an important point, that just as Linda Gale said, the participants understood that we're trying to do research to find a vaccine, and we know that this one didn't work. So I just like to put out our thanks to all for that. I think that we have a lot to learn yet from this study. I'll make a few comments, and then open it up, I guess, for questions, because I think these might be some points that, well, this is what some of the questions we receive is, what, you know, why was it that this didn't work, and where do we go from here? And one of the things I think, just as a very simple exercise that we need to do, is we need to make sure that the vaccine induced the immune responses that we expected it would. Sometimes you call that a disaster check, just to make sure that the vaccine performed as we had expected. Now, we don't have any reason to think it didn't, but that's just a standard kind of procedure. So in the laboratory, we'll be testing samples to make sure that the vaccine performed the way we thought it would. We'll also look to see at the types of viruses that some of the participants were acquired, exposed to, and then became infected with. And we would like to make sure that there's no real difference. This is sometimes called a sieve effect, where you look at the viruses that came across and were able to cause an infection in the setting of people who received a vaccine versus those who did not receive a vaccine. Sometimes if a vaccine had an effect, you can see a difference in the type of viruses that came across. Because the numbers are so equal, it's not clear that there would be any difference, the numbers of infections in each group, but we will conduct that study called a sieve analysis to see if there was any pressure at all on the viruses that people were exposed to. But there is a larger context, as Mitchell was saying, because this study, 702, Rhonda, was so closely linked to RV144, which is still, unfortunately, to date, the only HIV vaccine trial that ever showed any protection. And of course, the Rhonda was conducted with the hopes that we could even do better than the RV144 trial. And there were some immune responses in the participants in the RV144 trial that were thought to be linked to protection, or at least reduced risk of acquiring the virus. And we need to look at those specific immune responses to see if they were in fact induced in 702 participants. Because then we'll know that particular immune response, though it may have worked in RV144 in Thailand, it did not prevail in 702. So that's an important aspect of this study, is to understand if the same responses that were thought to protect in RV144 were present, then we'll know that those were not strong enough or the right type to prevent infection in the setting of South Africa. So what does this mean? Some people say, well, does this mean that RV144 didn't work? Or so I'm not sure what it means. I think what we can be confident about, of course, is that this vaccine did not work in South Africa. And I think we can be sure about that. There are a lot of differences between these two studies, although we tried to keep things similar. You're probably aware that the epidemics are very different. In Thailand, it was a very low-level epidemic, whereas in South Africa, it's a very much higher incidence rate of infection. It's a different type of virus, and some aspects of the vaccines were different necessarily to address that. So we didn't have an exact apples to apples comparison. And then I guess the other point I wanted to make is we often get asked, what does this mean for other prevention studies? Or does this mean, we won't ever have a vaccine? Or what about other vaccine studies that are happening? So we wanted to make sure, and I think as Linda Gale has explained, there's been a lot of effort to communicate. In fact, this webinar is a great example of that, to make sure we get the right message out there that this result is really restricted to 702. We don't have the results from the other, for example, large-scale vaccine study that's being conducted in South Africa and other parts of the world. So we have to wait for those results until next year when they're ready to become available. It does not generalize to other current vaccine studies. There are differences between those vaccine regimens, and we don't expect this result to convey due to the different types of vaccines. I guess the other last point I'll close with is that while there are, of course, there's widespread disappointment, and we're working with scientists and vaccine developers to plan what our next steps are. For example, like looking at the immune responses and to see if the viruses were similar between vaccine and placebo groups. We will be conducting other studies to try to help us understand why the vaccine didn't work, but we don't have that plan completely laid out yet. It's still under discussion. And so I guess I wanted to close to say that we have not given up on development of an HIV vaccine. We think we need an HIV vaccine now more than ever. And we have a diversified portfolio. We have other studies ongoing, and we have other vaccine strategies planned. And I could talk about those a little bit more, but I'll reserve those comments in case there's other questions. So that's all I have. Thanks.
Great. Mary, thank you so much. And I hope we do get back to that larger pipeline. And we will be sure in the follow-up, you actually did a phenomenal job of describing the full pipeline last May on a webinar around Vaccine Awareness Day. And we will be sure to post that. Obviously, the 702 result was unknown then, but you did a brilliant job, and we'll be sure to repost that link. But before we get to questions, I do want to turn to my friend and colleague Ntando Yola, who works both at the Desmond Tutu HIV Foundation with Linda Gale, and also one of the co-founders of APHA in South Africa, a fabulous new robust prevention advocacy organization, to provide some additional thoughts and also open up for some inputs from other community members in South Africa.
Thank you very much, Mitchell, and to Linda Gale and Mary as well for really that backdrop in terms of where we at and what it is that needs to happen. And I am sure we all appreciate the fact that for all the 4,500 participants who really have helped the field, as well as the communities that have been behind them to really bring us to this point. It is also very much important to hear from some of the people who've been to different extent working with them. So we have really brought along some of those members who are working in different parts of South Africa with these communities. We have Nombeko Mbongo, who is here in Cape Town in the Western Cape, working with us at the Desmond Tutu HIV Foundation. We have John Mdluli, who is in Johannesburg with Oram, as well as Miranda Mshongo, who is from KwaZulu-Natal in a town called Ladysmith, and as well as Blossom Makubalo from the Northwest. And I think at the end, we will also hear from a member of the Vaccine Advocacy Resource Group, who is Matthew Rose, to really also give an input in terms of these results. Now, really, one of the things as we're going to call to these participants is to, I think as Mary and Linda Gale have highlighted, is how important it is to really understand what the global community is saying, as well as the communities that have been taking part in these studies. And I think I will take no more time, but hand over to Nombeko, and then after Nombeko's remarks, I will hand over to the rest of the community engagement leaders to really share with us what it is that they are hearing on the ground and what are the messages that are coming through, and any other thoughts that they have. Nombeko, I hope you are, or you can unmute yourself and really give us a brief remarks.
Oh, okay. Hi, everyone. I'm Nombeko from Emma Vundleni site. Just to be brief, the participants and the community, they are actually disappointed that the vaccines came to this point where the study had to stop vaccinations, because as far as they say they hoped that because they participated for reasons like they would want to make sure that they put an end to HIV infections. And as much as they are disappointed, they are also clearly saying that they are hoping that there will be other investigations where they will be part of, and also there will be investigations about vaccines, so that we bridge the gap and we manage to slow down the number of infections, especially with males, because we had a little number of males in the study and we had more number of females, and males felt that this is one thing that they would participate in, and they would make sure that they engage in ensuring that there is low prevalence of HIV. And then on top of that, the communities, that is the biggest stakeholder engagement that we had sessions with, they showed a bit of I remember this session where there was quietness, and then the remarks were we're shocked. We thought that after the Thai study, this is going to lead us to where we say we are celebrating availability of HIV vaccines. And nonetheless, we understand that this is a research, and research can come up with any different outcomes. We are hoping that the researchers are not going to be tired and will keep on finding better other options of making sure that one day we do get HIV vaccines. Thank you.
Thank you very much, Nombeko. I think really the message is very consistent in terms of what is coming through from the communities. But let's hear from you, John. What is it that you have been finding?
Hi, Ntando, can you hear me?
Yes, John, very much clear. Thank you very much. Go ahead.
Yes, Ntando. The dissemination activities are currently happening in Gauteng as per the template that was submitted. But maybe just building up on what Nombeko has said, I think the news of HVTN 702 were received with great disappointment. So maybe looking at the responses or the reactions that we got from various stakeholders, I will talk about Ekurhuleni Ethics Committee. There were a couple of questions that were asked based on the presentation slides that we were given for use as a tool. One of the questions really that was asked was that 60% of participants have exceeded month 18. And the question was, why stopping the trial at 60% completion rate and not wait for all the participants to complete their vaccination visit to actually look at the impact? And I think the answer that was given was that it doesn't matter whether we stop it now or we continue up until the end of the trial, the results won't make any difference. As the data that has been collected to date has shown that no matter what we do moving forward, there won't be any different. And the second question that really came was that there were about 129 participants in the active arm who seroconverted, and 123 from the placebo arm. So they wanted to know, in terms of geographical spread amongst the 14 sites, where did we had a large number of infections. However, unfortunately, we couldn't answer this question. We had indicated to them that we will revert back to them. We're hoping that the final study results will cover the aspects of infection across different geographical areas. And then I think one of the issues that was really raised was the issue of VIPs. And also they wanted assurance in terms of VIPs services post-trial. And another question was they wanted to find out how long will this last amongst the participants who experienced this. And then they have also acknowledged the fact that some of the participants in the trial did fall pregnant. And chances are some of them might have been receiving an active product. And they wanted to find out if there are any plans to extend the VIPS services amongst the newborn children, especially whether the trial will have came to an end or not, looking at the fact that children might not have been participants in the trial. So they wanted to find out what are the plans to extend the VIPS services amongst the children who were born to trial participants who had VIPS. And maybe most importantly, in Tando, one of the issues was what are the implications of 702 amongst the currently existing studies. For example, a specification was made to studies such as HVTN 705 and HVTN 703. So the question was, what are the implications? Well, this outcomes or this news suggests that we stop 705 and 703, and we did give them an assurance to say the vaccine candidates were not the same. And I think that was the message we wanted to drive forward. And then lastly, I think this is the point that was raised to say when the whole study started, the trial or RV144 served as a basis for HVTN 702. And I think one of the concerns that was raised is what happened to that 1.2% that we had in the RV144 trial. I will end there, Rhonda. Thank you very much.
Thank you very much, John. And clearly, I guess as expected in times like this, that there will be more questions that really need to be answered. But let's move on fast so that we really leave time for more engagement with the rest of the global community. That is because we see that the questions are coming through. And I'll hand over to Miranda to hear if she has any quick thoughts, please, as well as Blossom.
Okay. Hi, everyone. Hello, Ntando?
Yes, Blossom, we can come in. Yes, we can hear you, Miranda. Thank you. Go ahead.
Yeah, okay. Yeah, I think John has covered most of the questions that actually came from our participants as well. So we first disseminated to the CAP, and they were also very disappointed from the results because they said that the community had hoped that we might have an effective HIV vaccine. And then, however, they appreciated, or they were happy to hear that the vaccines were safe. So there was nothing to worry about the participants from the side of safety. They actually thanked the site and the network for taking good care of participants because that was their main concern. And they said they were going to give support to the site by also disseminating the information to the community so that there are no myths or misconceptions. And then we disseminated to the participants because we actually had a meeting. We invited all participants to come for a meeting to the site on the 6th of February. The participants were very disappointed, but they were also happy to hear that the vaccine was safe. They also appreciated the fact that they learned, they appreciated the information that they got from their participation in the study, because they said they've actually learned lots of stuff from their participation. But they were contacted. I think what John has asked was the same questions that came from our participants. Their main concern was the issue of VISP. So they wanted to know, they said we should actually ask from the network. They want to know how long the VISP takes in person's body, how long is it going to last? Even if the study has to come to an end, are we still going to continue to actually look after them? What should happen? Can they now test at the clinics? So I think the main concern from them was actually around the issue of VISP. So yeah, I think that was the main thing that came from our participants. And then from the stakeholders, because we also had a meeting with the District 8 Council on the 13th of February, they actually said that they understood that we were trying. They appreciated the network and the site for actually trying, because everybody is working so hard in trying to actually find something for HIV prevention. But they were very disappointed because they said that they were also hoping that this vaccine might be successful, but they were also happy to hear that the vaccine was safe. So then they also said they were also going to give support in educating the communities so that we don't have the myths, because I think some of the members of the community were concerned that we were closing down. The study had come to an end. The site was closing down. So those are some of the rumors that we've been addressing in the community. But then they were also happy to hear about PrEP, that we're offering participant PrEP as well. So they also appreciated that. Thank you.
Thank you very much. Thank you very much, Miranda. It does seem like, important messages do need to go out to communities. Blossom, are you there to quickly, so that we really leave time to be able to have discussions with everyone?
Thank you, Ntando. I'll try to be quick. The immediate action from both communities and stakeholders, as well as our participants, was that of disappointment. Some of our community stakeholders, they were disappointed and they perceived the trial as being a failure. But it was important for us as a site to clarify that the trial was not a failure, because we don't want to leave the community feeling like the trial was a failure. It might then create problems in future if we want to go back and recruit, because they might see it as a waste of time, the trials are failures. So there we emphasize and zoom that the trial was a success. It answered the question that needed to be answered. It's just not the answer that we were hoping for. So after that explanation and education, they understood. But what also stood out was the level of disappointment by both community, CAP, stakeholders. It's the need for a vaccine. There's really that genuine need from all fronts in our communities that we want to see a vaccine one day. So I think we can, when we go out with future HIV vaccine trials, we can really leverage on that the communities want to be part of the answer. They want to take part, and it's evident in the disappointment in the community dialogues that I've had. The other concern that came out in the community was they were a bit alarmed that more participants seroconverted on the vaccine arm compared to the placebo arm, and we spend time because we don't want them, because that had to be handled very carefully and in a sensitive manner, because we don't want misconceptions. So in explaining that to the community, we then explained to the community, we re-emphasized that the vaccine in no way increased or decreased participants' risk of HIV. So we wanted to alleviate that fear and alarm. We then also touched on the HIV prevention package that we offered our participants when they came for the follow-up visit. Things like risk reduction, counseling, education, condoms, PrEP. So they were quite happy that we actually really took care of the participants. And the same as what Miranda said, they were also very happy that we offered the participants PrEP. That we actually, they even said, wow, you guys really care for the participants. It's not just a matter of us getting participants into the trials, but we also care, because the reason why we took time to explain that, because we don't want communities to feel like, wow, the test vaccine maybe placed our participants at risk, because that might be an issue. It might be an issue when we want to go back and recruit. And some of our communities also were interested in what care that we give the participants that eventually seroconverted. But then we then explained to them that we didn't just kick the participants out of the trial the moment we found out that they seroconverted. We still had follow-ups with the participants. We did counseling, we did education, and we also down-referred to the local facilities. And we followed up if they actually went after we referred them, because we want to show our community that we're really taking care of participants. And what also stood out for me is that when it comes to trials, it's ongoing education. We don't just go recruit, get accrual, and then just disappear from communities. We must always educate. There must be that strong foundation of education, so that when results like these come out, that our communities are prepared, that they can accept the results. And it also minimizes misconceptions when the results eventually come out. It's an ongoing process that our communities and stakeholders and participants have to journey with us during the trial. So that stood out for me, that we really need to escalate education in our communities. And one thing with our participants, some of them, they also asked the first question. They wanted to know, okay, can we now test at the facility? Can we don't have to worry about this anymore. We did education there. And some of our participants also, they were confused by the SMS. Some of them, when they received the SMS, they thought that they're no longer required to come back to the site. So I think next time when we do the SMS, we must be clear in the message of the SMS, because some of them, when they were called to come to site, they were like, oh, but I thought we are done. The SMS, they misunderstood the SMS. I'm trying to be sure I can think under
that's fine. That's a lot of work that is happening on the ground to really work with communities and limit as much as possible any misrepresentation of information and messaging. Thank you very much, Blossom. That was helpful. And Matthew, please, really just a few minutes, because not even a minute, we're running out of time, but a word from the VARG.
Yeah, I'll be quick. So the VARG has been closely watching this. We are excited with the level of community engagement, and that community has really been at the forefront in making sure that they get results, and that, science on this is always going to be challenging, but we can still design trials that will truly let community understand what's going on and put things in the context, in which why calls like these are so important. And I think we're also hopeful for the way forward. I think that from RV144, we have some places to be looking at, and we can now finally have a trial where we can actually examine those places to see if our assumptions about what we needed to look for were activated and happening. But also, we have a diverse set of strategies that allow us to look forward at some of the future trials that are also happening on the continent. And I'll leave my remarks pending there.
Thank you very much, Matthew. So yeah, I think we've really heard from all of those perspectives, and a lot of questions are coming through. And I think I will hand it over to you, Mitchell, to really guide us in terms of some of those questions and who takes the questions from the panelists.
Great, and Thando and all of you, thank you so very much. And I know we are toward the top of the hour. We will keep this line open for as long as people are able to stay, because there's so many important points that are raised. Perhaps I'll cluster a couple of things just off the top, and maybe first to you, Linda Gale. As you were speaking and you talked about the updated protocol with 12 months more follow-up, someone had emailed in a question about, in addition to participants hearing about the results themselves, what might you do differently with respect to oral PrEP, if anything? And you, of course, in addition to being a vaccine researcher, have also been a leader in PrEP rollout. And then related to that, a question came in about, do you know anything about PrEP use in 702, and whether for current other trials or future trials, with an incidence rate or the number of infections we saw, is there more that needs to happen within the context of trials of other prevention modalities to make best use of the things that already work, particularly oral PrEP?
Yeah, thanks, Mitchell. So let me start with the aspect that PrEP, frankly, uptake in 702 has been disappointing. But I think it's very important to put that aside, that PrEP uptake in other sites, other than demonstration sites, have also been somewhat disappointing. So I do think we need to contextualize this with the concept that PrEP is new in South Africa. It is uninitiated. It hasn't had the demand creation that one would have hoped simply because of where the country is. I think that is about to change. So I'm hoping that our increased efforts to promote PrEP at all 14 sites will coincide very nicely with much greater demand creation efforts from the National Department of Health as the big rollout of PrEP is about to undergo. So I think what we are doing at the site level is ensuring again that sites are fully informed, site level staff are fully informed about PrEP and how PrEP works. We will continue to make sure that there is PrEP availability at sites. And the MRC and others are working very hard to make sure we can keep our hands on oral tenofovir emtricitabine to make sure that it's always available at site or within easy reach of site staff. And as I say, even at the regional meeting next week, there will be a lot of conversation both about our participants who have seroconverted, just to check in how people are doing. We always do that. Check that individuals are well taken care of, that everything is moving along well in that regard, although, as was mentioned, I think by Blossom, all of those participants have been referred into the treatment program in South Africa. But on the PrEP side, because the national program is still young and not quite there yet, we will continue to provide parallel PrEP at the sites. We're putting out increased information, increased demand creation at sites, and of course the message will be, particularly amongst young women, there was a very high incidence rate, and this is an additional way that people can protect themselves, and they should make good use of it. So that message will be going out. In addition, other modalities, of course, VISP is very much our mandate. As long as people are followed up and beyond, we will continue to provide services for VISP. Luckily, we have not seen much of it in 702, but we will as part of our unblinding and going forward, we will again be checking to see who has VISP and who doesn't. And those people will be notified, and we will carefully manage them and provide services for them appropriately.
Great, no, super helpful, Linda Gale, and timely given, as you say, the PrEP scale up in South Africa. Kind of a bridge question both for you and I think also maybe separately for you, Mary. One is just that there's a lot of information obviously that no one has, and I think John, you raised the issue about the geographic differentials within the 14 trial sites. Lots of other additional information forthcoming. Linda Gale, do you have a sense of when additional information will be forthcoming beyond what you all have to date? And I think, Mary, you also mentioned some of the analyses. Are these things that we expect to hear over the next several months? Will it take a year or more? Just to give people a sense of timing.
Yeah, so I can already sort of give people indication that we have a broad dissemination plan at this point. So definitely the primary publication we've already started on. So, people can expect to actually just read the basics in terms of the primary endpoints and the primary analysis that led to this point. That we are working on that manuscript even as we speak. There will be a short intervention at CROI, which is held in Boston at the beginning of next month. And Glenda will be on a panel, again, transmitting details about the study there. We are then very much targeting R4P. R4P will be in October in Cape Town. And we would imagine, as Mary says, we're starting to map out what other immunological type analyses we could possibly be doing. And one would hope that by October, some of those results will be coming in. So I would imagine, people should watch out for convenings such as, in the first instance, CROI, perhaps some more conversation at AIDS 2020, but really focusing on R4P to hopefully have some in-depth answers to bring to stakeholders about what might have happened and what has been a reason for this outcome. I will say that clearly there are sub-analyses that will also happen, looking at gender and age and geography. So again, those results we expect over the next few months.
Great. That is great, and obviously we look forward to keeping all those conversations going, and I know everyone on this call clearly quite interested and looking forward to all of that coming out. Mary, picking up on one of the issues, and someone asked a question, you talked about the differences, of course, between the vaccine in 702 compared to the one in RV144, that it was modified. But you mentioned also the other large trial, particularly two trials right now, working with the Janssen vaccine, the HVTN 705 and HVTN 706 trials. And a question just, is there any relationship, and what might, why we expect it to be the 702 result doesn't imply anything about the 705, 706 result? And if you could just describe a bit about why those vaccines are actually different.
Sure. Okay, thank you. It's a great question. So the vaccines that are being tested in 705 and 706 do have a prime boost approach. So initially, the participants are vaccinated with one type of vaccine. There's different components, and then boosted with another. And that is similar to what was used in RV144 as far as the strategy, but the components of the vaccine are very different. In the vaccine in 705 and 706, not only are the types of vaccines different, the way they encode the messages to instruct the immune system are different as well. So in 705 and 706, the vaccine is thought to be able to be used globally. Whereas with RV144 and 702, the vaccines were made fairly specifically for the area in which they were studied. So, for example, in 702, Hambo, the vaccine there was designed to help protect against the circulating clade C viruses that occur in Southern Africa. But the vaccine design for 705 and 706 are called mosaics, where at least the thinking is scientifically they have enough information in them to possibly prevent infection for multiple different types of viral strains or clades. So that's one important difference, and you'll often hear people talk about the mosaic or the 705, 706 vaccine as a global vaccine. And that's something that's an important question for those two studies. Could the vaccine protect in one area better than other, or at all? So those are important questions. The other area of difference is the immune responses that the vaccines should elicit are different. The 705, 706 vaccine strategy activates other types of the immune response, so different cell types will be called in to help that could be considered antiviral or could be considered protective. And then the actual boosting, the latter part of the vaccine regimen is different as well. It's different design, and it's formulated differently with a different adjuvant. So there are enough differences between the two strategies that it's entirely possible there could be protection in one and not the other, because they are so different, and that was what made them worthy of being tested. So I think we'll just have to wait to see the results as they become available, hopefully next year, or those and later for the ongoing studies.
Mary, that was one of the clearest descriptions of how they're different and why that research is importantly going forward. As you were speaking, though, a question came in that I think is really important and would be great just to get your quick take on this. And we often talk about these go, no-go decisions when we go into a big trial. And a fabulous colleague wrote, were the go, no-go criteria for the 705 and 706 trials the same criteria as for 702, which were based on RV144? And I think we know the answer, but I just wanted to make sure you clarified that.
They were not the same. As I mentioned, there were some different, there are what we call the immune profiles. The immune profiles are different between those two vaccines. So the go-no-go criteria were not the same. There were anticipated to be certain antibody responses that could be directed at different parts of the viruses that may prevent infection, but they were not identical or the same. They were different.
Great. No, that's super helpful, and I think important for people to realize that while they are both prime boost approaches, they are very different, and we will follow the research. That is great. I realize we're past the top of the hour. Perhaps I will just turn to each of you very quickly to hear if there are any last thoughts. I know some people have had to drop off already, but maybe and Tondo, I'll start with you to see if you and then perhaps we'll go around to some of the other panelists here for any last thoughts before we close.
No, great. Thank you, Michelle. And I mean, I think this, whenever because this is really not the first time we really come across this kind of an outcome. And I think the very important message, especially in country in South Africa, is how it is important to really work together across all the fields, you know, from researchers to funders to communities, most importantly, and really galvanize. I think Mary put it very well, and I think Linda Gale as well, that, you know, it is when, you know, when we get this kind of outcome, it becomes even more important that we galvanize and solidify our efforts to work with communities and bring them on board. And I think it does seem like, you know, we have the communities with us, and as you know, with the help of everyone, including the advocates in different countries in the world, who are really, really passionate about this work and are really wanting to contribute in ensuring that we do find eventually the vaccine that we're looking for. So I guess I think it's the message really remains important, that you know, there's still more that is in the pipeline. There's still a lot of work that is being done by the field to really make sure that we recover from this. Even though, you know, we have been on this hope street, it seems like we've been detoured a little bit, but we do need to find a way to come back and continue being hopeful.
Great. And Thando, thank you so much. I wonder, Namboko, John, Blossom, Miranda, I can't remember the exact order, but any of you want to add some last thoughts?
Hi, it's Nambeko.
Hey, Nambeko, go ahead.
One more thing that came up is like one of the stakeholders raised that was in more of a layperson's language, was a person gets pregnant because being pregnant is a normal thing. And the expectation is to give birth to a healthy baby. And it sometimes happens that people do get stillborn. So that's how that stakeholder tried to comfort themselves. And they even said, in the sense of the HIV vaccines, there is no way that one cannot try again. And also, it's the same thing as when somebody is pregnant and getting a stillborn, it doesn't mean that you stop trying for another baby again. So they said the message that they would say to the researchers is that keep going.
Beautiful, Nambeko, thank you. Miranda, Blossom, or John?
Hi, it's Miranda.
Hi, this is Blossom.
Oh. Go ahead,
Miranda, you can go.
Blossom.
Yeah, no, from QM side, I think we also saying thank you to HVTN, and I think the way that we've actually educated our community made it a little bit easier when we had to disseminate these disappointing results, because they were aware that we were not promising the community that this vaccine was effective, even though they had that hope that it might be successful. But we were very transparent, even with the participants, we were very transparent, so they were aware of everything that was happening. I think that is why we had participants saying thank you to the network, thank you to the site for even making us part of this journey. And they say that they are still going to continue to support, even with, if we have other studies that will be coming up, they will still support the site and the networks. So we're also saying thank you to the network, because I think they actually tried. So, and as a site, we've also committed ourselves that we're also going to continue with the fight and the struggle.
Great, thanks, Miranda. Blossom.
Hi, from my side, I think as community engagement, we just need to keep on reminding our communities that HIV vaccine research is a journey, and we are in this journey together with our communities. And without our communities, we will not get to an HIV vaccine. So it's only by working together and by journeying together will we give an HIV vaccine, especially in South Africa. I could feel the disappointment. You could actually cut it with a knife, and it just shows that our communities are backing us, and it's only through working together. It's not only up to researchers, but it's the collective. We need to journey together to get an HIV vaccine. So that is a message we should keep on emphasizing in our communities, not to lose hope. This is not the end.
Great, thank you. And I guess John and Matthew, any last thoughts?
I just think that I want to echo what everyone else has said about having the community involved, but say what an exciting place it is that we are. Yes, we have a disappointing result, but to think that we are in a place where we have such big studies going on right after it, and we're able to dive right in on getting the information out there and trying to understand this trial while also informing other trials is a place we haven't been in quite some time. And that keeps me hopeful in this long kind of marathon it is to find a vaccine.
Great, thanks. And John?
And maybe John is on mute, but Mitchell, I also wanted to really highlight one of advocates who is with us, who actually has worked as well as much with the communities, Nandi, who is also with us on the call. And she might not be with HVTN currently, but is still really a big part of the work that you are doing. And perhaps it would be great to hear from her as well.
Love it. Nandi?
Thank you, Rhonda, and thanks everyone. So I now work for APHA as the Regional Stakeholder Engagement Advisor. But in my previous role with the HVTN, I was privileged to have been part of the incredible team that worked on engaging, educating, recruiting, enrolling, as well as retaining the 5,000 volunteers that participated in the Uhambo study. And I must say it was both an exciting and a challenging undertaking. And that is why it was heartening to learn that the vaccines used in the study did not prevent HIV infection. So although the views are devastating for all of us, I strongly do believe that through robust partnerships as advocates, researchers, industry, funders, government, and all other key stakeholders, we can cultivate this energy towards ensuring that our participants and communities fully comprehend the rationale to stop further vaccination. I do know that the HVTN is working tirelessly to make sure that this happens satisfactorily. And just to help complement some of the continuation activities planned by the VTN, I think it might be helpful for the HVTN to consider engaging with national, provincial, just all local stakeholders to help explain why the vaccines are stopped. This could possibly be done through existing forums such as Cross-National Stakeholder Engagement Forum. Also, engaging in rigorous community sensitization and education efforts on the different HIV prevention trials that are ongoing, such as HVTN 705 and 706, and those that are about to begin, like the PEDVAC study, is fundamental, as mentioned by Blossom and some of our other speakers. And this will help reinvigorate some of the support for HIV prevention vaccines and HIV prevention research as a whole in our community. We should also consider how to take the lessons learned from HVTN 702 and manage expectations, helping communities and participants understand that in research, we hope for good outcomes, but the scientific process means that we won't always get the results that we hope for. And it was great to hear from Linda Gill and other speakers that participants clearly understand this. I also strongly believe that the HVTN should continue working with sidecabs and advocates to gather intel on the ground to help dispel some of the myths and misconceptions that may arise about the studies, thereby paving the way for future vaccine prevention studies. And this is something that was also noticed by some of our speakers like Miranda from the Lady Smith site. Lastly, as advocates, we are availing ourselves and are happy to partner with anyone and everyone to make sure that our communities understand what transpired in the study. That while disappointing, while the results are amazing, it is so good that we have this information for the children, and that we need to continue to support HIV prevention research, as the vaccine is truly our only hope to finally end this epidemic. Thank you.
Great, Nandi, beautifully said. And maybe before I close, Linda Gale, any last thoughts from where you sit in Cape Town?
I think, Mitchell, everyone has said really great stuff today. I've learned a lot. I hope others have had many questions answered. I would just answer by saying that in this country, it really behoves us to continue to promote prevention in all of its forms. That which we have in our hand, that which we will have soon, and of course keep our eye on the ultimate prize of a preventive vaccine. And if we continue to work together like this, I think these bumps in the road will merely be the stories we pass down in history, and we will prevail. So I want to thank everyone again for their amazing solidarity.
Great, beautifully said, Linda Gill. Thank you. And Mary, I know it's still early on the West Coast, but any last comments from you?
Yes, I just wanted to thank everybody for participating, and also to thank you so much to hear the feedback from the participants. I hadn't had much of that feedback, and it was very helpful and very encouraging to hear that. So thanks.
Great. Thank you so much, Mary. And really to hear it from whether one of the co-chairs of a protocol, whether from one of the lead funders, and especially from people working at this site level, it really highlights what we always say and what we all know so well is that it truly takes all parties, all stakeholders to come together. And whether you are, no matter what your acronym is, APHA, AVAC, HVTN, DAIDS, VARG, CAB this, cite that, understanding that we all have a role to play and we're all in it together. And that is an important reminder in the best of times and in some of the more challenging times. I know we didn't get to all the questions, and we also know that there's so many more questions that will only come as data begin to come out at CROI, as Linda Gale said, at R4P in October, and into 2021 and beyond, not only from this trial, but from other trials. We look forward to working with all parties, all acronyms, all stakeholders in trying to together navigate through. And I think Linda Gale perhaps most beautifully said, focusing on delivering what we have today at scale and with urgency, while maintaining the unyielding desire and need to develop the next generation of options. And I think that's exactly right for all of us to take forward. We will be posting this recording and other materials as they come out from CROI and in the future. And if you do have more questions, please do send them directly to us. We will compile them along with our partners at APHA, HVTN, and VARG, and be sure that there is increasing amount of information so that we all can keep up to date and know what to do with this information. So again, I just want to thank everybody on the call who spoke, and all of you who stayed on for well past an hour and a quarter. And I know we have lots more to talk about going forward. Have great days, and look forward to continued conversations.
Thank you.
Thank you. Bye.
Bye-bye.
Cheers all.